Good afternoon to all the NEET aspirants. This is your Prasanna Ma'am from NEET World. So today we are going to discuss about the enzymes topic from the chapter Biomolecules. Before going into the concept, let's have a brief intro about NEET World special programs. For 11th standard students, we have NEET World's 2-step to medicine, where the fee is very minimal of 3333 per month, and for 12th standard students we have NEET World's 1-step to medicine program with the fee of 3333 per month, and NEET World's power step to medicine. This is exclusively for long term students, which is with the same fee. So in this same fee structure only, you will get the complete lecture content both in offline and online mode, and then you will also get the complete study materials with 2 rapid revision master revision strokes at the end, okay, up to the May month of 2026. Okay, so this is all about the programs beta. To know more about the programs or these courses, you can call us to the given number, or you can directly visit our branch which is in Narayanguda, Hyderabad. So let's get into the concept, that is enzymes, which is most important and crucial for our NEET exam. Definitely we can expect one question for sure, okay, one question though for sure we can expect from this enzymes topic. Today let us deal the enzymes. What is the difference between the organic enzymes, that is biocatalysts and inorganic catalysts, and let us also look into the factors that are affecting enzyme activity, mechanisms of enzyme activity. Let us discuss about all these concepts in today's session. So first, how can you define the enzymes? How can you define the enzymes beta? Enzymes are called as mostly biocatalyst, which will speed up the rate of reactions. Okay, what are enzymes? Mostly enzymes are proteinaceous in nature, or we can also call it as what? Biocatalysts. Proteinaceous in nature or biocatalysts that will speed up the rate of biochemical reaction, or they will speed up the rate of reaction. Fine, ma'am. So how can you define the enzymes as? Enzymes are biocatalysts which increase the rate of reactions, or we can also say like enzymes are proteinaceous in nature which will increase the rate of biochemical reaction. They will speed up the reaction, or we can also say they catalyze the reaction. Some nucleic acids will also behave like enzymes. Some of the nucleic acids will also behave like enzymes. Best example is ribozyme. Ribozyme is one type of nucleic acid. This enzyme will catalyze the reaction. Clear beta? These 2 points are fine, right? Enzymes are organic catalysts mostly. There are some inorganic catalysts also, but this inorganic catalysts do not occur in the living cells. This is a brief introduction about enzymes. First, let us look into what is the key difference between enzymes and inorganic catalysts. Enzymes, which are also called as biocatalysts, okay, these are what ma? Biocatalysts, and inorganic catalysts. Let us look into the differences properly. Enzymes are complex biological macromolecules. These are made up of protein. So they are what? Proteinaceous. Proteinaceous. Fine. Whereas inorganic catalysts are simple, small sized molecules or elements, you can call it as. Second difference, these catalysts will operate only within the living organisms or biological systems, whereas inorganic catalysts are found and they function in what? Non-living systems, that is outside the cells. Each enzyme is highly specific to single biochemical reaction. For every biochemical reaction, we have a unique enzyme. So there is high level of specificity in the biocatalyst. Here if you see, a single inorganic catalyst can act on multiple different chemical reactions, okay, inorganic catalysts. And here enzymes lose activity at temperature above 40 degrees Celsius. So temperature is affecting the enzymatic activity in the biological reactions. Whereas here in the inorganic catalysts, they remain stable and they work efficiently even at high temperatures and pressures. Even at high temperature and pressures, they are stable and they will work efficiently. This is the differences between biocatalysts and inorganic catalysts. Fine beta, these differences are clear, right? They are highly important. Take note of it, or you can take a screenshot of it also. And now you look here, as temperature is one of the factor and limitation of the enzymatic activity, here we have this point which says that enzymes which are derived from thermophilic organisms are thermally stable. Okay, what enzymes are thermally stable? Those enzymes which are derived from thermophilic organisms, and they retain their catalytic activity even at high temperatures. Those temperatures can rise up to 80 degrees Celsius to 90 degrees Celsius. Up to this high temperatures, they can continue their catalytic activity. Whereas normal biocatalysts, their optimum temperature will be 40 degrees Celsius. Whereas those enzymes which are derived from thermophilic organisms, they retain the enzymatic activity even from 80 to 90 degree Celsius. This ensures the thermal stability, which is the key feature of this enzymes which are derived from the thermophilic organisms. Clear beta, clear with this point, right? Now let us look into the mechanism of enzyme action. First you keep this in your mind. Enzymes will have some crevices, or we can say this as pockets. Okay, enzymes will be having some crevices or pockets. To that crevices or pockets, the substrate will come and bind. Okay, so substrate will fit to the crevices or pockets which are in the enzyme. Substrate enzyme complex will be formed here. Okay, so enzyme will be having what, Nana? Crevices or pockets into which the substrate will be fit. Okay, and this will form enzyme substrate complex. Okay, this will form enzyme substrate complex, and this enzyme substrate complex at the high state, that state is called as transition state. Okay, this is the transition state wherein the enzyme substrate complex will turn to enzyme product complex. Okay, enzyme substrate complex will turn to enzyme product complex, and that is how the products are released. Here you can see the substrate. What is the substrate here, beta? Sucrose, and the enzyme is invertase or sucrase, you can call it as. Okay, invertase. In this enzyme, to this crevices or pockets, you can say, in this crevices, sucrose, that is the substrate, will come and fit, forming out to be an enzyme substrate complex. And at the highest state, that is transition state, enzyme substrate complex will turn out to be what? Enzyme product complex, and this will show some change in their structure, and that is how products are being released. From what? Substrate is turning out to be products here, that is glucose and fructose. Okay, glucose and fructose. This is how, and this glucose in the respiratory pathway, this will enter into the process of glycolysis. Okay, so from sucrose, how glucose and fructose are formed, you have understood now, beta? Substrate, enzyme, and that is how products are formed from enzyme substrate complex to enzyme product complex. And what is the state called as, beta? Transition state. Till here it is clear, Nama? Now you see, you have understood the mechanism of enzyme action. Now you see there are two models that are proposed. First one is lock and key hypothesis, second one is induced fit model. Induced fit model. Let us look into both the models. First is what? Lock and key hypothesis. In this hypothesis, the scientist says that to this lock, the substrate will come and bind as a key. Okay, to this lock, substrate will come and bind as key, forming enzyme substrate complex. The enzyme is rigid, that it will not change its shape or structure. The active site structure will not be changed. Substrate will come and fit to the enzyme like a key fits to the lock, and that is how enzyme substrate complex is formed. This is being explained in what model, ma? Lock and key hypothesis. This you see in the induced fit model, that substrate can change its shape or size. Okay, the shape is changed or altered according to the enzyme active sites. The active sites of the enzyme can change their shape or structure based on the substrate that is binding to the active site, like how we fit the hand into the gloves. Okay, like how the hand is fit into the gloves. In the same way, the active sites of the enzyme, that structure is modulated according to the substrate. Clear, right beta? So as such, the enzyme will fit into the substrate will fit into the enzyme, and the substrate is converted to the product, and this is how the products are being released. This is induced fit model. Induced fit model is same like hands fitting into the gloves, and lock and key hypothesis is same like key fitting into the lock. Understood, ma'am? So let us look into the key differences between this lock and key hypothesis. Lock and key hypothesis and induced fit model. Let us look the key differences between these two models. Lock and key hypothesis is proposed by M.L. Fisher in the year 1892. Whereas induced fit model is proposed by Daniel Koshland in the year 1958. Okay, this is the primary thing, that lock and key hypothesis is proposed by Fisher and induced fit model is proposed by Koshland. Induced fit model is widely accepted. Okay, it is widely accepted because of this following characteristics. Let us look into it one by one. First, if you have to see beta, here enzyme's active site is exactly complementary to the substrate shape. So there is no change in the enzyme's active site in the lock and key hypothesis. Whereas if you see in the induced fit model, the active site shape of the enzyme will be changed, or it will be changed according to the shape of the substrate. Okay, depending upon the shape of the substrate, the active site shape will change from the enzyme. And here you see key is what? Substrate which fits into the lock, that is enzyme. Here the hand which is fitting into the glove, I said you, right? Hand is what? Substrate, and the glove we can imagine as enzyme. Flexibility of the enzyme. Here the enzyme in lock and key hypothesis is not flexible. This is rigid because it is not altering its shape according to the substrate, whereas here the flexibility is more. The enzyme is flexible, it adjusts the shape of the active site depending on the substrate. And look at here ma, specificity. Specificity here in lock and key hypothesis, the enzyme is highly specific. High specificity is seen due to the perfect fit. Here in induced fit model, moderate to high specificity is observed. Induced fit model, moderate to high specificity of the enzyme active site is seen. This will fit it, optimized after interaction. And lastly, you look into it ma, adaptability to the substrate. Here in lock and key hypothesis, the enzyme cannot explain enzymes binding similar to the substrates. Okay, because in the living reactions in the biological systems where the enzyme shape will be changed according to the substrate, but this is not being acceptable when we compare it with what? Lock and key hypothesis. But here if you see, this can explain, this model can explain binding to a range of similar substrates. This can explain to a range of similar substrates because enzyme is flexible over here. Okay, so which model is widely accepted? That is induced fit model. That is because of the flexibility of enzymes. Clear till here, Nama? Next. So now this chart is very important from your NCERT. Okay. So till here what we have learned about the mechanism of enzyme activity, wherein enzyme binding to the substrate will first form the enzyme substrate complex, and this enzyme substrate complex at transition state, this will get turned to enzyme product complex, and then products will be released. Okay, products will be released at the end. Clear, right ma'am? So here if you see, just a second. Yes, let's continue. So here products are formed at the end. In this enzyme substrate complex change into enzyme product complex is called as the transition state. Okay, enzyme substrate turning to enzyme product complex is called as transition state, which is the highest state of energy, highest state of potential energy, okay, wherein the bonds between the enzyme substrate are partially breaked and the bonds between enzyme product complex are partially formed. So here you can see partial breakage and partial breaking of enzyme substrate complex and partially the bonds that are formed between enzyme product complex, and that state is what? Transition state, which exhibits high level of potential energy. What is this activation energy over here? Activation energy is nothing but the minimal energy that is required to form the enzyme product complex or to attain the transition state. What is the activation energy, beta? The minimal energy that is required, to attain transition state. Fine, you got the clear difference between transition state energy and the activation energy na beta. Transition state energy is the highest level of energy where the bonds between the enzyme substrate complex are partially breaked and the bonds between enzyme product complex are partially formed. Clear, right? So here this is called as what? Transition state energy. This is the highest potential energy. Activation energy, you can define it as the minimal energy to attain the transition state. So in the presence of enzyme, the activation energy is very minimal. The activation energy with the enzyme is very minimal to attain this transition state. In the absence of the enzyme, the activation energy required to attain the transition state is too much high. Okay, it requires so much of the substrate. Okay, so this is about transition state energy and activation energy. Here you have to also know about exothermic and endothermic reaction beta. Okay, exothermic and endothermic reaction. So here if you have to look into this graph, on the x-axis what is plotted beta? On the x-axis progress of the reaction is seen, on the y-axis potential energy is observed. So the highest potential energy is what? Transition state, where enzyme product complex will be forming out there. So here if you look into it in this graph, substrate level energy is more when compared to the product range. Okay, so here product energy is less, product potential energy is less and substrate potential energy is high, which means that the energy is being released. Energy is being released in the reaction, so it is what? Exothermic, where the energy is released in the form of heat. Energy released in the form of heat. Fine. Endothermic reaction. In endothermic reaction, the substrate potential energy, if you have to compare with y-axis, the substrate will be minimal and the product will be high. The potential energy of substrate will be less in the graph and the potential energy of product will be high. If that is the scenario, it is endothermic reaction where heat is absorbed, in the reaction. Okay, heat or the energy is absorbed. Clear, right, ma? So here you have got clearly about the activation energy, transition state energy, what is exothermic reaction and what is endothermic reaction. This is a graph from your NCERT which is clearly showing you the exothermic reaction from substrate to product, wherein substrate potential energy is high and product is low, heat is released, energy is released in the form of heat. Clear? So this is exothermic reaction. Now here if you have to look into the factors affecting the enzyme activity. Very good morning, Lucky. Good afternoon actually. Good afternoon, ma. Factors affecting enzyme activity. 3 factors will be affecting the enzyme activity. First is what? Temperature. As we have previously learnt that biocatalysts, Okay, biocatalyst will show the enzyme activity, and the optimum range of temperature is what? 40 degrees Celsius. And those enzymes which are derived from the thermophilic organisms, can show the enzymatic activity above 80 degrees Celsius as well. So here we are talking about the normal biocatalyst, the enzymes which are acting as biocatalyst. So the optimum range of temperature will usually be what? 40 degree Celsius only. So the enzyme activity will increase drastically up to the optimum temperature, and you can see the drastic downfall of the enzyme activity or the rate of reaction once the optimum temperature level is reached. Once the optimum temperature level is reached, the enzyme activity or the rate of reaction will reduce. Clear right now? This is the first factor, that is temperature. Let us look into the second factor, that is pH. Okay, pH will be different for different enzymes. pH range also is optimal for each and every enzyme, and that is specific. Okay, so the optimum pH is specific for each enzyme, where the enzyme activity or rate of reaction will be increasing till the optimum pH is reached, and once the optimum pH is reached, then there will be a drastic downfall in the rate of reaction. So this is about the second factor that is affecting the reaction, rate of reaction or enzyme activity. Here on the x axis the factor will be given beta pH, on the y axis rate of reaction. Same the case of temperature, on the x axis temperature, which is the factor affecting the enzymatic activity, and on the y axis the rate of reaction is given. So here you can see, as I said you, it is specific for all the enzymes. These are few enzymes, biocatalysts, and this is its function and optimum pH. If you want to take, you can take a screenshot of this, as the function and location is already clear to us. Let us look into the pH range only here. So here if you have to see the first enzyme, that is pepsin, the optimum pH is what? 2.0, that is 2 pH. Next trypsin, its optimum pH is 8, and salivary amylase, its pH is 6.827, lipase 7.528, maltase, it is 6 to 7 pH, renin, that is also called as chymosin, which pH, optimum pH is 3.524, DNA polymerase, it has optimum pH of 7.528, catalase, its optimum pH is around 7 pH. So here proton concentration, the number of protons will also decide the rate of reaction or enzyme activity. Let us look into third most important factor that is affecting the enzyme activity here. Third factor is substrate concentration. Substrate concentration. So here on the x axis the substrate concentration is plotted, and on the y axis the reaction rate as usual. So as the substrate concentration is increased, the velocity of the reaction will be increased. First point, do remember beta, what is that? As the substrate concentration is increased, velocity of the reaction will be increased. Substrate concentration, which is indicated by Michaelis-Menten constant, that is Km, and Vmax is indicated as the velocity of the reaction. But once the substrate concentration is maximum, it is reached, once the substrate got saturated with the enzymes completely, then even if you increase the substrate level also, there will not be any change in the velocity or the rate of the reactions. Once completely the substrates and the enzyme get saturated, then even if you increase the substrate level, there will not be any increase in the velocity or the rate of reaction of the enzyme. And here I have to tell you one more point, the maximum level of the substrates, okay, that Km, as I told you, which is Michaelis-Menten constant, here this will give you Vmax by 2, Km is equal to Vmax by 2. Okay. At maximum substrate concentration, half of the velocity of the rate of reaction will be attained. Okay. Km is equal to Vmax by 2, and after this, even you increase the substrate concentration also, there will not be any increase in the rate of reaction. So, here with respect to substrate concentration, 2 points you have to remember beta. As substrate concentration is increased, initially rate of the reaction or velocity of the reaction will be increased. Second, Km, which is called as Michaelis-Menten constant, it is explained as Vmax by 2. At this particular position, half of the velocity of rate of reaction will be attained. So, these 2 points you just remember. In our next class, we will finish the part 2 of the enzyme. So, stay tuned. Fine. I would like to end the session by once again briefing you about NEET World special programs, that is for 11th, 12th and for repeaters. 2 steps to medicine, NEET World's 1 step to medicine and Power step to medicine, with the same minimal fee of 3333 per month, which is feasible for all the students belonging to different financial backgrounds. To know more about this courses, you can call us to the given number or directly visit our branch, which is in Narayanguda, Hyderabad. And lastly, I would like to brief you about our Telegram group, okay, NEET World's Telegram group, which is a public platform which is beneficial to all the students who are preparing by their own for NEET 2026. In this Telegram group, what do you get? You will get the daily quizzes of botany, zoology, chemistry, physics and mathematics for the people who are preparing for JEE. And in this group, you will get the live class schedules, timings also, and free resources of questionnaire books or study materials are provided in online. One-on-one doubt solving with faculty is provided. Any doubt anywhere if you drop it in the Telegram group, we will be solving you that doubt. And you can call us to this number if you are facing any challenges in NEET 2025 counseling might be, or how to prepare for NEET 2026 might be. Our faculty support will be there for you. How to log into this Telegram group? By just scanning this QR code or by visiting our official website and log into the Telegram group. With this, I will sum up today's session and we will meet in the tomorrow's session by discussing about enzymes part 2. Thank you.